Hello friends, welcome to my channel Pharma Gyan. Friends, in this channel you can learn about pharmaceutical guidelines, pharmaceutical industrial practices, processes, documentation practices, and many more about pharmaceutical industries. Friends, please don't forget to like, subscribe and share my channel Pharma Gyan, and press bell icon given in bottom right corner to get latest updates directly in your notification panel when I upload new videos. So next we will discuss about the video.
Active Pharmaceutical Ingredients Committee, A.P.I.C. Cleaning Validation Questions and Answers. Guidance on aspects of cleaning validation in active pharmaceutical ingredient plants. September 2016. Question 1. When should a company validate/revalidate cleaning procedure? When is validation not required? Companies should look at each situation individually and determine the need for validation. Section 7.0 provides a basic template, which may be used as a starting point in this evaluation. The necessity to revalidate cleaning procedures should be determined under change control parameters, see Section 10.0. If routine verification procedures are used, these should be monitored to ensure that the procedure is in control. Companies should consider a periodic evaluation of cleaning procedures, which are subject to variation, i.e. manual procedures etc., as an additional precaution to assure that the procedures are still valid. Question 2. When is it appropriate to use prospective, concurrent or retrospective validation? Retrospective validation of cleaning is not condoned by regulatory authorities. Prospective validation is the ideal method of validation. In situations where very few runs are manufactured in any given period and or a business decision has been taken to release the next material manufactured after cleaning based on a high level of testing of the equipment, i.e. validation level, concurrent release of material may take place. Question 3. What level of testing is needed after cleaning validation? The answer to this question depends on individual situations. Typically, companies perform visual inspection and take rinse samples to monitor the effectiveness of the cleaning in predefined intervals, time or number of batches. If after validation company decides to perform always cleaning verification, non-specific scientifically sound analytical methods may be used. A practical approach for monitoring the effectiveness of cleaning after completion of cleaning validation in an effective, scientific sound and inexpensive way is given below. 1. Visual inspection of the cleaned equipment. Only after this check is considered satisfactory, proceed with the next step. 2. Take a rinse and forward/or swab sample. 1 liter of rinsing liquid is usually required. 3. Determine the dry residue by evaporating about 500 milliliters to dryness in a small flask using a rotary evaporator. This unspecific test covers also inorganic salts, known or unknown organic products, and will detect the total residues. This test might be omitted for the drying equipment. In this instance we have a pure API or intermediate and typically no potential for side products, degradation, etc. 4. If the result meets the specification, proceed to specific, chromatographic, technique. Start with a TLC limit test, inexpensive and fast to validate. Broad detection range UV and specific derivatization. If these techniques are combined, the method is very specific for the different impurities potentially present in the sample. Apply 2 samples, the last washing liquid, to see all potential residues, the rinsing liquid, to look for the residue, and 2 standards. 1 of the suspected residual products at a concentration that is the limit accepted, and a 1:2 dilution of the standard. If the main spot in the rinsing liquid has lower intensity than the standard, the equipment is clean. The second standard is for confirmation of detection. 5. If TLC is not the appropriate technique, revert to HPLC or GC. Question 4. What critical parameters need to be looked at during cleaning validation? It is vital that the equipment design is evaluated in detail in conjunction with the product residues to be removed, the available cleaning agents and the cleaning techniques. Also, the ruggedness and reproducibility of the cleaning procedure should be covered. Question 5. What number of cleans should be run in order to validate a cleaning procedure? A validation program generally encompasses 3 consecutive successful replicates. However, companies should evaluate each situation individually. Question 6. Is it acceptable for a validated cleaning procedure to be continued until the analytical results demonstrate it is clean? Advice: regulatory authorities do not condone this practice. When the analytical result does not meet the acceptance criteria, an investigation to determine the possible root cause should be performed. If needed, retraining of the operators should be performed and / adjustment of the cleaning procedure to solve the issue. Question 7. Is it necessary for companies to validate a maximum time allowed for a piece of equipment to be dirty before cleaning? Advice: companies should have SOPs in place, which require cleaning to be performed immediately after production has stopped. This scenario should be validated. However, if for some reason immediate cleaning is not always possible, companies should consider the effect of time on the material deposited on the equipment. It may be possible to group or bracket products, and validate a worst-case scenario. Question 8. Is it necessary for companies to validate a maximum time allowed for a piece of equipment to be left clean before reuse? Advice: companies should have SOPs in place to ensure that pieces of equipment are adequately protected from any contamination after cleaning has taken place, i.e., ensure that the equipment is adequately covered, closed from dust, etc. If the company feels that there is any risk of contamination during idle time after cleaning, validation should be considered.